Hello everybody, let's have a look on neuroglia cells that occur in the central nervous system. First to be mentioned are the astrocytes. There are two types of astrocytes, or two phenotypes. First, there are cells with many processes but enough cytoplasm. These are called protoplasmic astrocytes. And they occur in the gray matter. Their end feet are touching and even embracing the capillaries in the central nervous system. So these are protoplasmic astrocytes. There's plenty of cytoplasm here, and they occur in the gray matter of central nervous system. They have processes called perivascular processes, or end feet, and they are surrounding the capillaries of brain or spinal cord. Another phenotype looks a little bit different and occurs in the white matter. There's a small cell body, long and slender processes, and they are called fibrous astrocytes. The processes are long, slender, and again they are surrounding the capillaries, but this time in the white matter, of the central nervous system. Well, the astrocytes in both gray and white matter are responsible for mechanical stability, mechanical support of neurons. They have intermediate filaments in their cytoskeleton that help to promote this stability, and the typical intermediate filaments are called GFAP, which stands for Glial Fibrillar Acidic Protein. And I'm afraid you will meet these intermediate filaments in some tumor diagnostics in pathology later on. They are part of the blood-brain barrier, and they form the layer called glial limiting membrane. There are these membranes in the human body. First, it's perivascular, membrane formed by these end feet and superficial glial limiting membrane on the very surface of the brain, for example. The second type to be mentioned is called oligodendroglia cells. These are producers of myelin in CNS. They produce the myelin in the central nervous system. Let's imagine several nerve fibers that might be axons or dendrites of various neurons, and there is the oligodendrocyte with its cell body, and long processes that resemble tentacles of octopus, that are wrapped around the fibers. So one oligodendroglia or oligodendrocyte has several processes that are wrapped around the processes of neurons. So axons, dendrites, and the inner layers of these processes are producing the myelin. Myelinization in the central nervous system is a necessary condition for some circuits in CNS to work. It starts with the 4th month of prenatal development, but it proceeds to the 2nd year and quite rapidly, but it could be proven up to the 10th year of age. So the myelinization in CNS is an ongoing process during human ontogenesis. The third type would be ependyma cells. Ependyma cells are lining the CNS cavities. They have cell junctions in between. They're usually columnar, they have microvilli, and they have long kinocilia. They are arranged similar to epithelia. And there are special modifications called tanycytes that have their end feet around the capillaries. So if this is the CNS cavity with cerebrospinal fluid, and this would be the brain or spinal cord. This is one of the barriers between these two. We got the cell junctions sealing the spaces here. Between the ependyma cells. This is ependyma. that's kinocilia and microvilli. This is a special type called tennicide. For example, in the third brain ventricle, with its perivascular feet surrounding the capillaries. So as such, ependyma cells are part of the encephaloliquor barrier. Which is the barrier between the cerebrospinal fluid on this side and the brain or spinal cord tissue. The fourth type would be microglia cells. Microglia occur usually in the resting form as quite inconspicuous cells with long nuclei with some slender processes. Microglia do not originate from nervous system. They differentiate from the circulating monocytes. So blood monocytes are entering CNS where they become microglia. This is how resting or quiescent microglia look like. However, as a part of inflammatory reaction, they might be activated. And then they become macrophages, tissue macrophages. Yeah, with their phagosomes, lysosomes. Where they can phagocyte, so they have the ability of phagocytosis. The respiratory burst. And presentation of antigens. Therefore promoting other part of immune reaction in brain or spinal cord, namely the specific immune response. So please note that the microglia are the only neuroglia that do not originate from the CNS, but they migrate in later on.